So thank you very much, Andy. I have to say it's an excitement to be here, to presenting to you my first experience in this, what are they calling the brave new world. And to start with, I have to say that I come actually from the old world. I'm based in Porto, in Portugal, on this research institute here, which is the largest research institute in health in Portugal. And within that research institute, we do have a diagnostic unit where molecular pathology actually takes a big chunk of the activities that we do there. One thing that is extremely important for us is the quality of the work that we do, and we search our laboratory accreditation both at the CAP level for some of the tests, but also at the new ISO 15189 and the ISO 17025. And we actually just got news that we are the first accredited labs for both tissue and liquid biopsy testing in Europe. And our work with NGS really goes back to the early days, as Andy mentioned, almost in 2010 when the PGM came. We were the initial lab to perform bracket testing using the ion PGM by then. And during these years, we've been just evolving our capacity, both at the sequencing level, at the bioinformatic level, but also at the templating capacity to increase the number of tests that we do. And it was during this summer that we got this huge package back in our facility, which actually it contained inside the first Genexus apparatus that is just here in the lab together with my first ion PGM, just for you to see the reference in size between the two systems. So since August, we've been sort of playing and stressing a little bit the system to see how it would handle in real life facilities. And what we did was initially to use commercial control samples, both Horizon and Seracare, FPE and cell-free DNA reference material, where we just pushed it through the system. And this was also a learning curve for us to see how the software and all this information would be shown up to us. As you see here, the ship now is broken into 4 different lanes that can be used individually. And this is actually the result for the first samples that we run in the system. And as you see here, the division on the 4 lanes, we almost reached close to the 60 million individual reads that were obtained from this single run. As was stressed, this is all the way from sample to report, and this is the variant report that we got from the system. This was important, again, by using control samples that we could see that we could both detect single nucleotide variants here at different allelic molecular frequencies here, but also different fusions, and also copy number variants that could be detected all at the same time from any given sample in the system. What further then we did was to use our clinical research samples and now really using real life samples from in this particular case, lung cancer tissue and liquid biopsies that we had before previously characterized using the systems that we had in the lab to compare how they would behave in the new system. So very quickly, what we got was we ran a series of samples, FFPE samples, both RNA and DNA components. These samples had been, as I said, previously characterized by our S5XL system using colon and lung and the comprehensive panel. And what you see here is that the different samples that were tested harbored different driver mutations that were actually all detected at similar levels with the new system, but due to the different coverage of the 2 panels, we did indeed get a broader spectrum of alterations that we could detect now in the samples, combining the Genexus with the Opa assay. What was curious was that one of the variants that was detected with the new system on this particular sample was the T790M that for any reason was not detected previously. I do suspect that it could be that the new assay has a broader robustness so that it can provide a better calling for these variants, but I could not, of course, exclude any intrasample heterogeneity because in this particular case, I could not use the same DNA sample because it had been previously fully spent out on the previous characterization. But where I think things really pushed my happiness in the lab was when I started to test the liquid biopsies. And this is one of the samples that I did. And for you to have an idea of how the system can really get into routine testing, this was a sample that was started to be prepared at 9 a.m. in the morning. So we spent a couple of minutes just to process the plasma, to centrifugate the plasma and push it through the total nucleic acids isolation. And here, this was, we still do it manually, so it was really quite a lot of hands-on time to process the sample. At 4 p.m., we went to the Genexus system, and 10 minutes hands-on, we had the samples being sequenced and prepared and sequenced. And the next morning, when I got back into the lab, I had a full report completely characterizing this sample. This was a sample of a lung cancer plasma sample at recurrence. So we could, the combination of the system with the precision assay detected the transforming mutation and also the resistant mutation that was causing the recurrence of this patient. So this is a prototypic system that can clearly take from biological sample to report with less than 24 hours. So I really do think that this will be a game changer. And to finalize, because my time was short, just some key words that I think are really making the difference with the system. First of all, it's an all-in-one solution. That is, it goes from nucleic acids to variants and to report all in one machine with minimal hands-on. It's really accessible because you have to know nothing about what next-generation sequencing is. Actually, my student that was there in one of the pictures, she had never been using any of the system, and she was actually brought into so that she could be one of the first trying out to see if she could get the machine working without any experience. And yeah, it was as simple as that. Actually, what one needs is a single pipette so that we can pipette the sample into the plate that goes into the sequencer. It has a fantastic thing that actually Andy did not refer, but I think it's one of the greatest things. It has a vision system, and the vision system is a set of cameras that really control everything that you are doing on the machine. So if by chance you just place one of the reagents in the wrong slot, it will tell you that it's not on that slot, so you have to change it. And it will continuous control if you are placing things on the right place and the correct amount in the system. So nothing goes through the system if it's not properly set up. And as I showed you, it can be a real fast instrument. And in the particular sample that I showed you on liquid biopsy, I got from plasma to report in less than 24 hours. So I do truly think that this system allows a wider implementation of NGS for genomic profiling, potentially bringing what we call precision medicine really closer to the clinical practice. So with that, I just finish up to bring you the first experience from this new world. Thank you.